Welcome to an introduction to the Genome Browser by Nextcode Health. Log on to the Clinical Sequence Analyzer using your email and password. Click on Studies and select the study for analysis. Once the study opens, you can click on Open Study in Sequence Miner. You will be prompted to launch the Sequence Miner Advanced Analysis Java module. Sequence Miner will open with a Genome Browser window open with BAM, variation, and coverage tracks already selected. Only 20 levels of reads are initially shown. You can double-click for all reads to be shown. If you right-click and go to Track Properties, you can choose between viewing reads rendered as left and right pairs or as forward and reverse strands, and whether the color shades increase with alignment quality or base calling quality. The track list on the left describe the tracks that are available in the viewer on the right. The upper track list and track viewer represent the subject-specific tracks in your study, like BAM tracks, variation tracks, and coverage tracks, while the lower track list and track viewer represent public reference data and tracks specific to genomic position. You can view dbSNP and ClinVars and even open up a concurrent UCSC browser in the track view. You can select which tracks are active in the track list by clicking in the box in the Active column. Highlighted in yellow in the track list are the tracks that are currently visible in the track viewer. Hovering above a track in the track list highlights it in the track viewer, and clicking on it brings it into view in the track viewer. You can right-click over the track viewer to move tracks up and down and generally manipulate the tracks available. You can right-click over a track and choose to view the data in a tabular format, because with the Genome Browser, you always have access to all of the data you are visualizing in a tabular format. For example, you can right-click over Ensemble Genes, click on View in Table, and instantly pops up a table with all the ensemble genes for the region delimited by the track viewer. Or, if we hover over this variation track and right-click and click View in Table, and you have all the variants for this individual for the range delimited by your window. You can drill into the report when it's appropriate. You can sort by column, filter on column, and when you found the gene you are interested in, you can sync the gene symbol or the position in the Genome Browser. You can float the track list by clicking this button, close the window to incorporate it again. You can select your position in the Genome Navigator using the chromosome graphics and the numerical position in the track viewer. You can zoom in or out, pan left or right, or scroll left or right. You can dial in 1 kb, 10 kb, 100 kb, 1 megabase, the whole chromosome or the whole genome to view. You can numerically dial in the genomic position of interest. For example, copy and paste from a UCSC browser window. Shift mouse scroll zooms in and out. Hit Control and you can use the mouse to scroll along. You can search in the genomic position dialog, and it opens a results window and you pick the result you intend to navigate to. If you want to add subjects to view in the Genome Browser, go to the File Explorer and open the Subject Reports folder, and then Distinct Participants. A list of all subjects you have access to will open, and you simply right-click and click on View Genome Tracks, and you can choose to open BAM tracks, coverage tracks, or variation tracks in the Genome Browser window of your choosing. You can also open tracks from the File Explorer under Studies, open the study you are interested in, and double-click on Tracks, and a new Genome Browser window will open with the subjects in the study you have chosen. Your first encounter with the Genome Browser might occur when performing analysis in the Clinical Sequence Analyzer. An interesting variant is encountered, but with your prior experience you know that there is no substitute for looking at the raw data. In this case, the Clinical Sequence Analyzer has discovered a de novo mutation, but are you convinced? Simply click on the variant or gene and it will automatically sync with an open Genome Browser or prompt you to launch the Sequence Miner. As you can see, the reads providing evidence for the de novo mutation are of high quality and provide unambiguous evidence for this variant as a causative mutation. Hopefully this brief introduction to the Genome Browser will help you get started in utilizing the many features of this powerful tool. Look for more training videos at nextcode.com.